Hello everyone, welcome to Tade MedChem Insights. I am Dr. Tade, Assistant Professor of Medicinal Chemistry. In this lecture, we explore the Prodrugs Design in detail. Don't forget to subscribe if you are a newcomer. Leave your valuable comments, like and share if you like the content. Let's start with the introduction part. The term prodrug was introduced by Adrian Albert in 1958, signifying compounds that require biotransformation to exhibit therapeutic activity. So we can define prodrug as biologically inactive, the one that can undergo bioreversible derivatives of drug molecules that undergo an enzymatic or a chemical transformation in vivo. For what purpose? Just to release the active drug which can exert the desired pharmacological effect. Initially, prodrugs were considered the last resort in drug development. However, advancement in combinatorial chemistry, high-throughput screening, and molecular biology have positioned prodrug as a central strategy in early-stage drug discovery. So, this shift has been driven by the ability of prodrugs to optimize physicochemical properties, overcome biological barriers such as blood-brain barrier, and achieve site-specific drug delivery. So that prodrugs were initially found serendipitously, with little knowledge at the time about the biochemical pathway that lead to their activation. With advancement in the field of pharmaceutics and drug metabolism, these processes were addressed and rational approach currently to the design of prodrugs were implemented to circumvent the pharmacokinetics and pharmacodynamic limitations. So, prodrugs are commonly designed to improve the pharmacokinetic properties like solubility, absorption, stability, target specificity, or formulation issues, particularly for oral administered drugs, and also designed to improve patient acceptance, reduce drug toxicity, and also for the slow release of drug in the body. So generally, when considered prodrugs, almost about 10 to 14% of drugs are approved globally that can be considered to be a prodrug. So in both drug discovery and development, even though the starting point is the one that can consider they are going to be used for drug development, and lately the rational approach considered that the products are important in the early stage of drug discovery. So in both cases of drug discovery and development, products have become an established tool. For what purpose? Just for improving the physiochemical property, the biopharmaceutical property, and the pharmacokinetic property of pharmacologically active molecules. So that is the important aspect of the prodrugs. Generally, the objective of prodrugs includes improving membrane permeability, so to pass the barriers so that they can become lipophilic, so they can increase water solubility, they can mask unpleasant taste if drug, for instance, is bitter, specifically for children. They also reduce irritation at injectable site and also enhance stability. Now, let's see classification of prodrugs. Before dealing to the classifications, let's see how the product is going to be arranged. So we have a drug, for instance, so that these drugs will not cross the barrier. So that in order to pass the barrier, we need a pro moiety. So that this pro moiety is going to be synthesized with that of the drug so that we can obtain the product. So this totally, the one that contains the drug with a promoiety through covalent bond can be considered as a prodrug. So these promoiety are temporary, the promoiety are temporary, and they should be non-toxic carrier groups. They can carry the drug to pass the barrier, so that after passing the barrier, by the help of enzyme or the chemical transformations, so the promoiety will leave the drug so that we can obtain the drug to its target. So that products are broadly classified based on their activation mechanism or structural features into two parts. So these are carrier linked products and bioprecursor products. So that the carrier linked products are drugs that are going to be linked through covalent bond to that of the promoiety, which we call it the carrier, so that it can be cleaved by the help of enzyme, or they may undergo chemical transformations into the active compound. So the active compound is going to be released to the target. In the case of bioprecursor, so there is no carrier. So there is no carrier is going to be there. So this is inactive molecules as always to the other products. So that can result from molecular modifications. Through what? Through oxidation or reduction. So rather than they can carry the carriers. So just to clarify simply, so we have a carrier that we call it the promoiety, which is non-toxic, and it is temporarily attached to the drug. So we have the active species. The active species are the one that can be considered as a drug, so they can be synthesized, so we can obtain the product, carrier product. So by the help of an enzyme or chemical transformation, so generally they undergo metabolism, so that can make the molecule to release the active species or the drug into the active target site, and the carrier drug should be also left, so that it should be non-toxic and it is temporarily attached to the drug. So let's come to carrier linked prodrugs. Carrier linked prodrugs are usually inactive and sometimes less active compounds formed by covalently linking a drug with its promoiety. The promoiety are temporarily, they are non-toxic carrier groups. So these carriers are often designed to have a strong affinity for specific biological targets, so that they can enable the product to bypass biological barriers such as cell membranes, for instance, or to home in on disease issues like tumor, so that the carrier temporarily masks or alters the parent drug physiochemical property, thereby improving solubility, stability, or pharmacokinetic profile of the drug. So they are formed by combining an active drug with the carrier species to form a compound with a desired chemical and biological characteristics. For example, a lipophilic moiety are going to be added to the drug to improve the transport of the drug through the membrane to pass the barriers. It can be add lipophilic. So the lipophilic moiety is a carrier. So the introduction of the carrier into the structure of a drug to form a product may be used to improve the pharmacokinetic profile of the molecules, or the solubility, or the stability, or the bioavailability, or in some cases it had used to direct the drug to specific areas, so they can enable them to have a site-specific delivery. So the link between carriers, that means the promoiety, and the active species must be a group such as esters, which can be easily cleaved by enzymes, esterase enzyme for instance, amide, it can be cleaved by amidase enzyme, so they can be easily metabolized be easily absorbed, and so that it can reach to the target. Or sometimes also the drug has been delivered to the required body compartment. That means the product can also enable the drug to be a site-specific delivery. So once the product reach the intended site, the linkage between the carrier and the active drug is totally cleaved, typically through enzyme or chemical transformation, so that they can release the active compound to the target. So this depends on the system actually. So either the molecule becomes inert or biologically active carrier moiety might be there. What is the requirement that has to be fulfilled by an ideal carrier associated product? So here we are going to see a well-designed carrier product satisfies the following criteria. These are the criteria or the requirement that should be fulfilled by an ideal carrier drug to be designed. So it involves temporary linkage. Don't forget, temporary linkage of the active molecules with the transport moiety that is frequently lipophilic in nature, such as polyethylene glycol or fatty chain or macromolecules, which increase membrane permeability and drug stability. For instance, the polyethylene glycol is a highly hydrophilic, non-ionic and flexible polymer, widely used to modify the pharmacokinetic and pharmacodynamic property of drugs, particularly those drugs which have low water solubility. This is the first criteria. Another criteria is the linkage between the drug substance and the transport moiety are usually binded covalently, but should be broken in vivo. That is another criteria. The other criteria is the product should be less active. Actually, usually they are inactive compounds as compared to the parent drug to be transported safely. Another criteria is products as well as the transport moiety should be released in vivo and must be non-toxic, so that the breakage of the linkage at the target site should be rapid and selective. They should release the active drug rapidly and should be selectively bind to its target, so that it can yield an effective therapeutic level of the active drug while minimizing alternative metabolic pathways, so that they can avoid toxicity due to the carrier molecule. What are the common carrier linkages? Let's take examples. Let's start from ester. So esters are going to be used for carboxylic acid or hydroxyl containing drugs in the case of enalapril. The other is amides. So amides are often used to improve stability in the case of valacyclovir. Another is phosphates. So these are going to be used to increase water solubility, specifically for parenterally administered drugs. Others are carbamate or carbonate. So these are commonly used to link amine or hydroxyl groups. So let's see a lot of functional groups that are going to be used to link carriers with drugs. Considering these groups are the drugs and these parts are the carriers that are going to be linked with the drug, and these are the possible examples. So here we have alcohols or phenols as a drug, so they can be linked through ester carrier linkage. So just considering here R can be alkyl or phenyl or other molecules can be there. In the case of amines, all types, that means primarily, secondarily or tertiary, or including also quaternary amines, or we have also imides, and also amides are going to be there. So the carrier linkage is amide. So where the R group can be alkyl, phenyl, or any other molecules can be there. In the case of aldehyde and ketones, we can use the carrier as carbamate, imine, or acetals. So the R groups here that are going to be joined might be aryl or alkyl groups may be there. In the case of carboxylic acid, so the carrier may be ester, dominantly ester, or sometimes imine is also there. So the R group here that can be joined to the carrier might be aryl or alkyl molecules is going to be attached to the molecule. So this is the way how the carrier linkage is going to be linked with the drug so that the product is going to be produced. The types of carrier linked prodrugs. So carrier linked prodrugs can be classified based on their linkage or based on their activation site. Based on their linkage, they can be classified into two parts, that is bipartate prodrug and tripartate prodrugs. So based on the activation site, we have two types again. There are type 1, which is known as intracellular, or type 2, that's known as extracellular. So when we come to the bipartate prodrugs, these are carriers is going to be linked or attached directly to the active drug. So that means the functional group is directly attached to the drug. What kind of functional group is going to be attached? Such as esters, amides, carbamates, carbonates, or phosphate is going to be linked with the drug. In the case of tripartate, as the name indicates, they are tri, so there are three groups are going to be there. One is the carrier, the other is the drug, the third part is the separator. So that the carrier is going to be linked to the drug by a link consisting of a separate structures. So the carrier is going to be connected in the case of tripartate to the active drug through distinct linker or spacer that are going to be used to directly link it, that can cause steric hindrance or stability issue. If there is stability issue or steric hindrance, we are going to produce a product as form of tripartate. So in the case of classification of carrier linked product based on their activation site, as we have said, they have two types, type 1 and type 2. So type 1 is intracellular. So when the target is in the intracellular receptors, so we are going to produce a product as form of type 1. So this is activated inside the cell, such as, to take example, antiviral nucleoside analogs are going to be produced in such a way. The other is extracellular, so this is activated in the gastrointestinal fluid or systemic circulations. Such products are going to be used to improve the absorption of the drug. In the carrier linked product system, the carrier is going to be removed by an enzyme controlled metabolism process. That is mostly seen in the bipartate case, and the linking structure that is going to be seen in the tripartate case, it can be removed by an enzyme system or it can be removed by the chemical reaction. Just to see examples how they can be connected. So this is an example, tolmentin glycine prodrug that can be converted to tolmentin, which is an acid. So this tolmentin glycine product is going to be designed to reduce the gastrointestinal side effect of tolmentin. So this is a bipartate example. So this is a drug. So the drug is going to be there until here. So as you have seen that the OH is going to be modified to amide form. So this is amide form. So the NHCO part is amide form. So the tolmentin is going to be changed to prodrug form with that of glycine, so that the amine part of the glycine, so because in the case of the glycine they have the NH2 part at the one end, so this is going to be connected with the OH part of the tolmentin. So this OH part of the tolmentin, it can be correlated with that of the glycine, so that they can produce an amide bond. That is the bipartate case. So this bond is going to be cleaved. The bond that's going to be found in the case of tolmentin is going to be removed as a form of enzyme controlled metabolic process. The other example is for the case of tripartate. So in the case of tripartate, there is a drug, so there is a carrier. In between the drug and the carrier, there is a linker, so that's a spacer. So that's the way they can be produced. For this example, bacampicillin is a prodrug for that of ampicillin. So in between the carrier and that of the drug itself, there is a linker. So that's the way we named as tripartate. So there are there are three important molecules. So the carrier, the linker, and the drug is going to be there. Now let's see about the application of carrier-linked prodrug. It's obvious that carrier-linked prodrug in drug discovery is one of the key strategies to overcome unfavorable drug properties. It's obvious that carrier-linked prodrug design is going to be produced by coupling the carrier moiety with active drugs. So there are a lot of applications. So the major applications that are going to be utilized using carrier-linked prodrug design is it can be used to improve the lipophilicity of a drug. It can also enhance the aqueous or water solubility of the drug. It can also increase metabolic stability. It can also improve patient acceptance. It can also improve site-specific delivery, specifically in case of barriers, so like that of blood-brain barrier. It can easily penetrate or can be targeting tumors like that. And also it can minimize side effects. We'll see each application with example in detail. Start the application of carrier-linked prodrug design with improving the lipophilicity of a drug or improving the permeability or bioavailability of the drug. So this is a kind of masking polar groups such as carboxylic acid or hydroxyl group is going to be masked with using lipophilic carriers, so that this lipophilic carrier will improve or increase the lipophilicity of the drug so that it can improve passive membrane permeability. So let's see examples that are going to be used to improve the permeability and bioavailability of the drug. So one of the example as a prodrug is the epinephrine XL. This is a kind of product for epinephrine, so that this drug will increase lipophilicity as compared to that of epinephrine because it can increase its corneal absorption and can be used for treatment of open angle glaucoma. As you have seen the structure of epinephrine, this is epinephrine, so it contains OH group which is polar, so this polar group should be masked so as to increase the absorption of the drug, and hence pivalic acid is going to be added so that between the OH and the COH part of the pivalic acid an ester bond is going to be produced. So this ester bond which are found in the prodrug can be easily cleaved by an enzyme known as esterase. So the esterase can be easily cleaved, so it can convert to the prodrug form of the molecules. When you come to the glaucoma is a group of eye disease that damage the optic nerve, often due to high fluid pressure, that is intraocular pressure, causing irreversible vision loss. So generally there are two main types of glaucoma that are named as open angle and angle closure. So the open angle is going to be happen gradually and it is the most common. In the case of angle closure, it is sudden and emergency. So actually there are also other types that include normal tension or secondary that can be caused by other conditions, and congenital is also another types of glaucoma, but the most common are the two, the open angle and angle closure. So the open angle glaucoma is the most common form of glaucoma, causing progressive and irreversible vision loss, usually in the peripheral, due to damaged optic nerve from high eye pressure. So it is a silent disease with no early symptom, caused by a slow chronic blockage of the eyes drainage system. Other examples include pivampicillin. So this is a pivalol oxymethyl ester. So this is the ester part or the lipophilic or the carrier that part that are going to be added. A pivalol oxymethyl ester part of the molecules. So this is the prodrug for ampicillin. So this is the one that can enhance the oral bioavailability of ampicillin because of its greater lipophilic characteristics as compared to that of the ampicillin. So other examples will include tenofovir, as we have seen. So in the case of tenofovir disoproxil, it is an important antiviral agent. So this is the lipophilic group that are going to be added as a carrier in the case of tenofovir disoproxil. So these molecules are going to be used for treatment of chronic hepatitis B and also for prevention and treatment of HIV-1. So it is given as a form of fumarate, as you know it. So fumarate is going to be added to tenofovir disoproxil to create a stable and pharmaceutically acceptable salt. That's known as tenofovir disoproxil fumarate, or abbreviated as TDF. That enhances the drug's solubility and also chemical stability and absorption in the intestine. So another example is enalapril. So this is enalapril. So it is also a prodrug that can be converted to enalaprillate. So it can be added here. This is a lipophilic group that is going to be added, monoethyl ester. So this is a monoethyl ester is going to be added. So it is a very good drug for angiotensin converting enzyme inhibitor. So they can be used to treat high blood pressure. Sometimes also it can be used for treatment of diabetic kidney disease and also heart failure. The other applications of carrier-linked prodrug includes enhancement of aqueous or water solubility. So this is critical for improving bioavailability of a drug and drug absorption, which can be achieved by reducing molecular hydrophobicity. That means the lipophilic nature of the drug is going to be reduced, or by introducing polar functional group such as a molecule that contains nitrogen or oxygen, or lowering crystal lattice energy. So what is crystal lattice energy? Crystal lattice energy is the measure of the strength of forces that can hold ions together in a solid crystal form. So what does it determine? It determines properties like hardness, high melting point, and low solubility. So primarily influenced by higher ion charges and smaller ionic radii. So what are the key strategies to enhance the water solubility or aqueous solubility? There are a number of key strategies. So one is modification of the molecule with their chemicals. So the chemical is going to be modified. Chemical modification is one important thing. So using different techniques. So in the case of chemical modification, or sometimes named as structural modifications, so we can add a lot of things. For example, we can introduce polar groups so that we can replace carbon atoms with polar heteroatoms like nitrogen and oxygen. Another way of chemical modification or structural modification is decreasing lipophilicity. So this is the one that can be used to reduce the overall hydrophobic character of the molecule or the drug. So this must be balanced with the need for membrane permeability. That means the balance between hydrophobic and hydrophilic nature should be balanced. And the third chemical modification is using salt formation. So salt formation is also another way that can be used to modify the structure. So this salt formation converts drug molecules into salt forms to improve dissolution. So the other strategy to enhance the aqueous solubility of a drug is physical method. So they can use a lot of approach. So for example, they can use particle size reduction. So particle size reduction is one way of improving the aqueous solubility using physical method. So this particle size reduction, you can use techniques like micronization, nanotechnology, or high pressure homogenization is going to be used to increase surface area. The other kind of strategy that can be used to improve the aqueous solubility is solid dispersion. So this is the one that can disperse hydrophobic drugs into solid carriers to enhance dissolution. The third part of strategy that are going to be used to enhance solubility is formulation techniques. So they can use a lot of things like cyclodextrin, for example. Cyclodextrin complexation is the one that can be used to improve the aqueous solubility. So this is the one that can be used cavity containing molecules to encapsulate hydrophobic compounds so that they can increase the solubility and bioavailability. Actually, there are also other way of formulation techniques that are going to be used to enhance aqueous solubility. For example, another is co-solvent. So utilizing mixture of water and water miscible solvents such as ethanol, this can be used to enhance also the solubility. The other formulation technique to improve aqueous solubility is pH adjustment. So, or it seems buffer system. So using buffers to maintain an optimal pH, which can dramatically increase solubility. So let's take examples that are going to be done as a product, carrier linked product that was improved their aqueous solubility. So water insolubility of prednisolone can be overcome by forming at carbon 21 of the drug. So this is the position at carbon 21 so that we can improve the aqueous solubility of prednisolones by using prednisolone 21 hemicyanate sodium. So this is the one that is very important to make the molecule to be aqueous soluble and so that they can be useful for parenteral administration. As you know that prednisolone is a corticosteroid or a steroidal hormone that can be used to treat certain types of allergies, inflammatory conditions, autoimmune disorders, and cancers. And also it can be used for electrolyte imbalance and also a skin condition. Another example is the fosamiperenavir. This is antiviral drug. So it's a prodrug that can be converted to that of the ampinavir. So this is also the one that can be used to enhance the water solubility of the drug. So ampinavir is going to be produced by phosphate ester modification. This is a modified part of the molecules. So the phosphate ester is going to be modified. So this is a sulfonamide prodrug of HIV protease inhibitor. Other drugs that are going to be enhanced, it is aqueous solubility is Solubility, which is a prodrug. So it can undergo two types of metabolism, that is reversible and irreversible kind of metabolism. So the one that undergo the reversible metabolism is the one that can give the active form of the drug that is going to be converted to Solubility sulfide. So this is the way that can be converted. So this group is going to be changed to the sulfide form. But the sulfur form, this is a sulfur form, is totally inactive. The other applications of carrier-linked product design is to increase metabolic stability of a drug. So this is the one that can be used to enhance compounds' resistance to enzymatic degradation. So some drugs are going to be easily degraded by the enzymes that are found in the liver, cytochrome P450 enzymes, so that this technique can help to improve the half-life of the drug, to improve the bioavailability of the drug, and also to reduce dosing frequency. So what are the strategies that can be used to increase the metabolic stability? One and the important strategy is to modify the structure, so chemical modification or structural modification of the drug. So in such case, we are going to replace metabolically susceptible group like methyl groups, like methylene groups, with more stable alternative like fluorine, deuterium, or saturated ring systems to prevent rapid degradation. We do also have other techniques or strategies that can be used to increase metabolic stability, such as bioaccessory replacement prodrug approach. So when it comes to the structure modifications, we can use or can introduce deuterium, that is the isotopic form of hydrogen, or we can use cyclization, or we can replacing metabolic soft spots that are going to be validated by in vitro assay like liver microsomes. So these metabolic soft spots are specific labile chemical bonds, often carbon and hydrogen bond, that are easily broken down so that it can lead to rapid metabolic clearance. Hence, it can bring poor bioavailability and it can make the molecule to have short half-life. So replacing metabolic soft spot is one of the essential medicinal chemistry technique that can be used to improve a drug's stability and efficacy by identifying and changing specific parts of a molecule that are rapidly degraded by the enzyme, that is by the cytochrome P450 enzymes in our body. So that is the important part of the replacement of metabolic soft spots. Let's see examples. The anti-leukemic agent that's known as cytarabine, or named as cytosine arabinoside, undergo a fast metabolism, so they can be changed to the inactive form of the drug, that is uracil arabinoside. That means here the amine part is totally deaminated by cytidine deaminase enzymes, so they can be changed to a ketone form. So the drug becomes short in terms of plasma half-lives. So how we are going to improve this fast metabolism of cytarabine? So we are going to use a prodrug that can be used to improve the metabolic stability of cytarabine. So the product that can be used is known as inositarbine. So this inositarbine is a prodrug which can be prepared to prolong the plasma half-life of cytarabine, and then it can also increase the concentration at the site of its activity. So this is the one that can be modified. So we are going to use fatty chain so that we can produce an amide bond here. Amide bond is going to be produced from the fatty chain. We can have the COH part, and here we have the NH2 part. So the CO part and that of the NH2 part is going to be binded together to produce the amide bond, so that the inositarbine will prolong the plasma half-life and concentration of cytarabine. The other application of carrier-linked prodrug is improvement of patient acceptance. So improving patient acceptance in drug discovery is achieved by shifting from traditional research-driven model to patient-centered and also co-creation model that can involve patients from the earliest stage of drug development. So this approach ensures that treatments address real-world need, so they can reduce burden on participants and also foster trust, so ultimately leading to higher adherence and better market uptake. So to take as an example, odor and taste are important aspects of drug administration. If a drug has a poor odor or too bitter taste, this is going to be rejected by patients, specifically in children. So furthermore, if a drug that can cause pain when it can be administered by injections, this is also the one that can determinant effect on patients. So this thing should be improved. So let's take example like that of chloramphenicol that can be converted to its palmitate ester form. So this is the one that can be used to minimize the bitter taste. So it is going to be palatable in pediatric liquid preparation form as form of suspension. So this is the normal drug, the chloramphenicol. So it can be added fatty chain, so palmitate is going to be added here, so that the drug is going to be easily palatable. So the bitter taste of the chloramphenicol is going to be vanished. So any kind of patient can easily take the drug without any kind of compliance. Let's include another example for improvement of patient acceptance, taking acetic acid. So the corrosive effect of acetic acid hinders its topical application as a fungicidal agent to treat fungus, so that this can be overcome by using a prodrug as a form of glyceryl triacetate ester. So this prodrug is going to be produced by esterification of glycerol with 3 molecules of acetic acid, so that the glycerol triacetate ester is not corrosive and can penetrate the skin more than the parent compound does. So this is the parent, that is the glycerol part, so that these are the acetic acid part of the molecule, so they can be converted to the acetic acid form in our body. So that is the way they can be used to improve its corrosive nature of the molecule, so that the patient's compliance is going to be improved. The other important application of carrier-linked prodrug is improving site-specific drug delivery. So this kind of technique is going to be focused on increasing local drug concentration at target tissue while minimizing the systemic toxicity. So what are the key methods that are going to be used to improve the site-specific drug delivery? So the key methods that include is using biocompatible nanoparticles, or using peptide-based drug conjugation, or using pH-responsive polymers, and using artificial interagent-driven drug design. So this is a currently used method to navigate biological barriers and also enhance efficacy in cancer, infectious, and inflammatory diseases. So this technique enables to target drugs to specific site in our body. So targeted drug delivery aims to concentrate medication in specific tissue while reducing exposure in other target. So there are 2 important strategies that can be used to improve site-specific drug delivery. These are site-directed drug delivery and site-specific drug release. So what does it mean? Site-directed drug delivery is a kind of strategy that involves directing the therapeutic agent specifically to its site of disease or injury, often by using carriers to guide the drug to its pharmacological receptor in protective form. In the case of site-specific drug release, this approach focuses on releasing the active ingredient only upon arrival at a particular location or in response to a specific trigger. So what is the goal of them? The goal of site-directed drug delivery is to minimize premature degradation and reduce systemic and also off-target effect by ensuring the drug reaches the disease area in higher concentration. So this is the one that can be act as kind of transportation system. So the site-specific drug delivery goal is to maximize the local therapeutic impact and control the timing of drug availability so that it can reduce the need for high and frequent doses. So in general, what is the difference between the site-directed drug delivery and site-specific drug release is that one is based on their mechanism of targeting, the other is their focus, and these are the two important distinctions. So in terms of mechanism of targeting, the site targeted or the site directed drug delivery rely on passive, structural, or physical targeting, such as the enhanced permeability and retention effect in case of tumors. While that of site specific often implies active targeting where the drug specifically binds to the receptor. That's the thing. In terms of focus, the difference is that site-directed focuses on the place. That means either the organ or the tissue, whereas site-specific focuses on the target component. So that is the cell or the receptors. But also they have similarity in terms of goal, in terms of mechanism, and in terms of application. So when we see the similarity in terms of goal, both strive to enhance the therapeutic index to get more effective with fewer side effects. In terms of mechanism, both frequently use carriers like nanoparticles or liposomes or polymers. In terms of application, both are used in cancer therapy, in inflammation, and gene therapy. Let's compare the two approaches, the site-directed drug delivery and the site-specific drug release, in terms of their focus, in terms of their method, in terms of their approach, and in terms of their primary goal. Let's start from the focus. The case of site-directed drug delivery, the focus is location and transportation. So how to get there, how we are going to deliver the drug to its target. In terms of site-specific drug release, it deals with timing, so the release timing. So when and where to trigger the drug. So when it comes to the methods, so the site-directed drug delivery might be active or it might be passive. But in the case of site-specific drug delivery, it is triggered by different conditions like that of pH or temperature or enzymes. So when it comes to the approach, the site-directed drug delivery is a carrier-focused approach. So they can use nanoparticles or liposomes. But in the case of site-specific drug release, so the approach is formulation-focused. So enteric coating system is going to be. So the case of site-specific drug release, it is formulation-focused, whereas the case of site-directed drug delivery is carrier-focused drug delivery system. So in terms of their primary goal, so the case of site-directed drug delivery is protection and accumulation. So they will protect early degradations. Whereas the case of site-specific drug release is it can bring precise release of the drug to its target. So let's see example for site-specific drug release generally. So very good example is the sulfasalazine. This is the one that can be used as a colon drug release by incorporating of drug into azo linkage. So this is a drug, as you have seen, this is a drug. So because of azo enzymes, so the azo reductase is the one that can cleave the nitrogen bond, so it can produce two drugs, sulfapyridine and aminosalicylic acid is going to be produced. So the azo linkage is going to be broken in the colon by the action of azo reductase enzyme, so that they can produce two different kind of molecules which are active. The aminosalicylic acid, the one that can be used for treatment of inflammation. The sulfapyridine is the one that can be used as antibacterial agent. Actually, we do have a lot of example for this site-directed drug delivery and site-specific drug delivery cases. To mention some of them for site-directed drug delivery, for instance, liposomal doxorubicin is one of the examples. So this is a kind of liposomic or lipid carrier which can be used to encapsulate doxorubicin. So they can enable it to accumulate in the tumor tissue through the enhancement of permeability and retention of the drug that are going to be used as treatment of cancer, so that they can reduce side effect. Another example is albumin bound paclitaxel, which is the anti-cancer drug. So this can be used albumin nanoparticles to deliver paclitaxel directly to tumor sites. We have also another drugs like ligand mediated targeting. So this is the one that can utilizing agents like folic acid, which can bind to folate receptors so that they can overexpress on cancer cells to deliver drug-loaded nanoparticles specifically to cancer cells. We can also have magnetic targeting drugs, for example, using iron oxide nanoparticles. So these are the ones that can be directed to tumor sites by an external magnetic field. For the case of site-specific drug delivery, we have also a lot of drugs that are going to be used. For example, for site-specific drug delivery include mosalamine. This is used for inflammatory bowel disease. So this drug is formulated to release specifically in the colon or terminal ileum. Another example for site-specific drug release is insulin-loaded aquasomes. So this uses a ceramic core coated with polyhydroxyl oligomers to protect the protein so that they can allow for targeted and sustained release. Another example is methotrexate liposomes. They can be used to treat localized and targeted treatment for arthritis. So that are some of the way that we are going to differentiate between site-directed drug delivery and site-specific drug delivery. We have a lot of drugs on market. The last applications of carrier-linked prodrug for our case is minimizing side effects. So minimizing side effects in drug discovery is achieved through different approach like rational drug design, high throughput screening, and precision medicine. So this will make the molecule to target to specific receptors while reducing off-target interaction or off-target effects. What does it mean by off-target effect or interaction? An off-target drug effect occurs when a medication binds to an intended protein or receptors in the body rather than it is intended target. This unintended binding often causes adverse, sometimes also unexpected and potentially harmful side effects. But we don't throw out the drug. So we are going to take the drug for therapeutic repurposing. So we do have a lot of key strategies to minimize side effects, such as nanotechnology based drug delivery or developing prodrugs. We'll give some example for this case. Structure based molecular modifications are going to be used. So this kind of technique or strategies can be used to enhance selectivity and to predict computational toxicity. So let's take for our case here a prodrug. So how a prodrug will minimize the toxic side effect of a drug, just taking salicylic acid. So salicylic acid is one of the oldest analgesic known. So this is the one that can cause gastric irritation as a side effect, and if in some cases if the things are adverse, it can bring bleeding. So that kind of side effects of salicylic acid will be reduced by converting the salicylic acid to aspirin. So aspirin is a product for salicylic acid. So this will be done by acylation. So this is the acyl part. So when we are going to add the acyl part to the molecule, this is the acyl part of the molecules, just removing the hydrogen part of the molecules, so we can obtain the aspirin. So aspirin a little bit reduce the side effect of salicylic acid, but still aspirin also have some kind of things. So adding acyl group is the one that can improve absorptions and also reduce the degree of stomach irritation. Still there is irritation, but it can reduce the degree of irritation. So since aspirin is mainly converted to salicylic acid by the enzyme known as esterase after absorption from the gastrointestinal tract. So this reduce the amount of salicylic acid that are going to be in contact with the gut wall lining. Let's come to the second types of products. As we have already said, that products can be categorized into two parts. One is carrier-linked product, the other is bioprecursor. So let's go to the bioprecursor. So these are unique class of products that are internally converted into active drug molecules by metabolic mechanisms. So what's the basic difference between carrier-linked product and bioprecursor is that in the case of bioprecursor products, there is no carrier parts. But in the case of carrier-linked product, for both bipartate and tripartate, they contains carrier parts. So, but here in the case of bioprecursor, there is no carriers. So thus the drug is intended chemically modified into an inactive biochemical precursor of a drug, which converts to the active drug in biological system. So this method can be especially useful for regulating lipophilicity, stability, and organotropism of a drug. What does it mean by organotropism? So this refers to susceptibility of a drug or chemical substance, what we call it pharmacon, to distribute to or concentrate in or exert its primary effect on specific organs or tissues. So this phenomenon is crucial for improving therapeutic efficacy and reducing systematic toxicity by targeting specific areas. So the bioprecursor product is generally activated by enzymatic reactions such as oxidation or reduction or hydrolysis. Just let's take example. For example, the anti-cancer Tade is an orally administered N-tetrahydrofuran derivative, which is a product of 5-fluorouracil, which is a fluoropyrimidine. So this is the Tade. So this is one of the important product that can be easily converted to 5-fluorouracil. So it can be converted primarily by the cytochrome P450 enzyme, subtype cytochrome P450 enzyme like 2A6. So this is the primary enzyme that can convert Tade to 5-fluorouracil. So enabling tumor selective activation, and therefore it can reduce systemic toxicity that are going to be associated by that of 5-fluorouracil during cancer chemotherapy. Let's see another example for bioprecursor products. As I have already said that this kind of products is going to be converted to its active form either through oxidation or reduction or hydrolysis. As we have seen earlier, the Tade is going to be converted to the 5-fluorouracil through enzyme-dependent oxidation. Here also we are going to see the cytotoxic agent cyclophosphamide is another example that can be converted through oxidation and also spontaneous hydrolysis. It can be converted to the active nitrogen mustard, or which call it phosphamide mustard. So this is the active form of the drug. So we start from the cyclophosphamide. So through different kind of oxidation and then hydrolysis, they will produce the active, or they will release the active molecules to the target. So let's start here. The cyclophosphamide through microsomal oxidation, so they will undergo, or they will add, hydroxyl group is going to be added, and sometimes can be oxidized by enzymes, so they can be changed to keto form for ketocyclophosphamide going to be formed, or it can be converted to aldophosphamide. So just the bond between the nitrogen and the OH is going to be this bond is going to be broken down, so they can be converted in such a form so that this is going to be totally hydrolyzed to give the OH part. So that is active molecules by releasing acrolein. So this is the active form how the molecule is going to be produced. Sometimes also it can be converted to carboxylic acid when aldehyde dehydrogenase is going to be act, but the active form is the nitrogen mustard. So the phosphamide mustard is the active part that can be used for treatment of cancer. What is the advantage of designing a drug using bioprecursor prodrug? So a bioprecursor prodrug is especially useful in overcoming certain pharmacokinetic and pharmacodynamic issue. So it has a lot of advantage. To mention some of the advantage of designing a drug using bioprecursor prodrug includes to improve bioavailability of drug, to reduce toxicity, and to increase the stability of the drug. When we see the improvement of bioavailability, so this bioprecursor is the one that can avoid first pass metabolism or low absorption. This is specifically for orally taken drugs, so that they can lead to increased plasma level of the active substance. In the case of reduction of toxicity, they allow for tissue-specific activation. As a result, they reduce side effects. Examples, as we have seen, are Solubility and Tagafor are very important examples. For example, Solubility is an important NASAID. So this is a bioprecursor design drug. So it can be changed to its active sulfide metabolite, and reductive activation by gut bacteria leads to less gastric irritation than other observed traditional non-steroidal anti-inflammatory drugs. So in the case of increasing the stability, it can protect the active drug from early degradation and further decomposition prior to storage or after release into the body, so that this can help to maintain its pharmacological activity. So this is the end for today's lesson. Thank you very much for watching this video, and also thank you for joining me here at Tade MedChem Insights. If you enjoyed with this lecture, hit the like button. It's really help to grow the channel, and share this with your friends, and don't forget to subscribe for more deep dives into the science of drugs, and also for more video breakdown. Until next time, stay curious, and I will see you in the next video. Bye.